Thank you so much, Professor Lo, for for the nice introduction. And I will try to. Oh, we are at the somewhere halfway, so I have to switch to the first slide. Otherwise, you already see the whole presentation. So then there's no surprise anymore. Okay, so today I would like to introduce you in wastewater treatment developments in the Netherlands, and also how we try with wastewater treatment to come to this circular economy. My name is Merle de Kreuk. I'm from TU Delft, from the Department of Water Management in the Faculty of Civil Engineering and Geosciences. So if we have economic activity, well, if I look outside, I should not be distracted too much by the beautiful view above your head. But if we have economic activity, we have waste, and generally waste will lead to environmental problems. But actually, we could also see waste as a mixture of undefined resources. And that's also why I don't like to talk about wastewater treatment, but actually we should see our wastewater as water with resources. These resources, we can get them out with biological processes. And then we can discuss if it's circularity or recovery, but it doesn't really matter, it's just in the name. So there are all kinds of considerations for recovery, and I have an example of phosphate recovery that we do now a lot in the Netherlands. It gets pretty normal actually, and all the water authorities would like to make a clean natural fertilizer out of our wastewater, so recover the ammonium and the phosphate and bring it back to the fertilizer, the artificial fertilizer industry, and well, it is a really nice circular thought, but on the other hand, we also solve a problem with it, because this phosphate in our equipment is really precipitating and clogging up our equipment. So we find struvite production in all the pipelines and in the reactors where we don't want it. So now we actually go into the recovery in the treatment plants and at the same time create a very nice product. So that is, yeah, it's two sides of the same coin. We solve a problem and also we can be more sustainable. So all the things that we could recover from a sewage treatment plant are actually in this slide as far as we know now. Maybe there's much more that we are not aware of yet. So if I run you through here in the activated sludge system, we could make bioplastics. There has been a pilot in the Netherlands to do so, and depending on the technology you could also extract alginate like exopolymers. I will tell a little bit more about that later. From the water we have, of course, the effluent, if we treat it well enough, we can get a lot of water back. The example was already mentioned. Also energy, we can create energy, but we should also reduce energy in the whole treatment. And then my own main interest at the moment is what we all can get from our sludge in the end. Sludge is so rich in nutrients and in energy that we should try to get everything out in a sludge digester. Well, and then there's a specific one, our toilet paper that comes in the form of cellulose. And in the Netherlands, we use about 13 kilos of toilet paper per person per year. I don't know about Hong Kong standards, but it might be more or less the same. So this cellulose we can recover and turn into paper again, but a pizza box from used toilet paper is perceived not too well. So what we could do with it is turn it into volatile fatty acids, and these volatile fatty acids we can turn into bioplastics again. So there we could also take a resource out and use it as a resource to make another product. Well, and as I said, I focus mainly on these digester systems. On the other hand, I would like to first start with one of I always mention it as my first child before I became a mother, the Aerobic Granular Sludge system, which is actually addressing this exopolymer production, water production and reduction of energy use. So the Aerobic Granular Sludge technology, I started to work on it in 2000 and was able to work on the development until the first full-scale installation when I shifted topic, but still I have some projects in the field. I would like to show you The Hague, the city where I come from in the Netherlands. Looks very different than Hong Kong, but still we have quite a few people living there. And we have a sewage treatment plant that's recently built. It's called Sewage Treatment Plant Harnaspolder, a very nice name abroad. But before, the sewage treatment plant was somewhere in the city and there was no place for expanding, so it was decided to move the whole treatment plant outside, build a whole new treatment plant, and then kind of conventional, so it was occupying a lot of space that we also can use somewhere else. And then my promoter, you might know him, Professor Mark van Loosdrecht, he's also very active here. He thought, well, we keep growing these bacteria in flocculated structures in sewage treatment. Wouldn't it be so much nicer to grow them in granules? Because granules will settle much faster and then we can get rid of all these clarifiers. So that's actually the idea he had, and the first reactor was there when I started working, and then we brought it to a new technology. And how does this work, making this aerobic granular sludge that looks, how can we get the bacteria to leave their nice flocculated environment where they have always enough oxygen and food, and kind of force them to grow in these aggregates? So that was the challenge that we had to figure that out, how we could do that and in a stable way, and that it also could stay like that. So actually there are a couple of tricks. What we managed to do is, with biotechnology, select the right organisms, and then the organisms that can have the initial uptake of the BOD into their cells during feeding. So that's one of the things that we do. And then during aeration, we let these cells grow on their internally stored substrate. So on their cell fat, as I always say to the students. And then every cycle there is a selection applied to all sludge to keep the granules, so every organisms that's sticking to their aggregate, they can stay in this nice system with plenty of food that we call wastewater treatment. And all the granules that misbehave and grow in flocks, we kick out. So that selection also helped to maintain these granules in the system. So there we transform the biomass structure. And then hydraulics comes in as well, because that is this selective wasting of the slower settling biomass. And so that's the way that the sludge is wasted that we kick out. So it's a combination of biotech and hydraulics in this system. Well, how does it work then in the end from a scientific point of view? Is that there is an oxygen gradient over the granule during aeration. So the food is like stored everywhere, but the oxygen concentration is maintained a little bit low and it is consumed in the outer layer. So we have an aerobic zone in these granules on the outside. And then in the middle it is anaerobic, but there's no oxygen because it cannot reach the core of these granules. Then during feeding, as I said, we had BOD storage in the cells of the organisms. So the whole granule is fat, they all have enough food. So in the outside they can use their food with oxygen, and in the inside they don't have oxygen, but in the outside there's nitrification going on. So the ammonium is nitrified to nitrate, and the nitrate can enter the center of this granule. So in the center of the granule, with the stored food, we can have the nitrate reduction to the nitrogen gas. And in the same time, since we make use of phosphate accumulating organisms, there's phosphate removal going on. So actually for the wastewater treaters here in the audience, what we do in all the different tanks in a normal UCT or biochemical phosphate removal system, we do here in one granule. And that was kind of a breakthrough. So, oh, it stopped. Oh, yes. So, and we have slow-growing organisms at the same time because we work with these phosphate-accumulating organisms. So, yeah, they also like to sit nicely together. They don't have the velocity to go out of this granule and escape and grow in weird filaments. So we have heterotrophic phosphate-accumulating organisms on the inside that denitrify, and we have ammonium oxidizers on the outside of the granule. And this is actually a picture that I took long time ago, where you could see that these ammonium oxidizers are on the outside and these heterotrophics are through the whole granule. Well, then that is done in practice, and this is a typical view of a cycle that you could see come across if you operate such a sequencing batch reactor. So you have a feeding period, and then you switch on the aeration, and you see an enormous peak of ammonium because that was all fed to the system, and of phosphate because it was all released from the organisms. And then during the aeration phase, that is nicely taken up. And here you see that there's a little bit of nitrate produced, but way less than the ammonium that's consumed. So that is the simultaneous nitrification, denitrification that's going on in these granules. And then there's a denitrification phase, and then everything nicely goes to very low concentrations that reach the effluent standards. So this, since 2012, this is the first full-scale installation was then built in the Netherlands, in EPA. And then it was also called Nereda, so maybe you know this granular sludge under this brand name. And it saves space, since it has a small footprint, because you don't need all this final clarifier. Then it also saves building materials, a lot of concrete, because you don't need to build these structures. It saves energy, because we don't need to pump around all this sludge, and everything happens in the same reactor. And it produces biopolymers, and that is one nice thing that can be recovered from this sludge. It is now produced under the brand name Kaumera, but it's actually a kind of alginate-like structure, a polymer that you can use in all kind of applications. And since 2012, there are over 60 of these reactors built. And that is really nice if you work on a PhD and in the end you really see it in practice. And there is actually a pilot now. So I was here in Hong Kong, and then I saw this technology also tried here in Hong Kong, which made me feel really proud. It's really nice. So this is this Nereda system. You can see it. This is in the north of the Netherlands. And this installation is treating half of the sewage coming in, and this whole installation is treating the other half. So there you can see that it's really compact. And also this one uses about 50 to 60% less energy, and the effluent standards are pretty nice. So for this treatment plant in Harnesporer, we don't need the final clarifiers anymore. This granular sludge concentration in the reactors are higher than we can reach with activated sludge, so we use also less space for the biological tanks. We don't need pre-treatment, it can all be just fed to these reactors. So in the end it uses 75% less area. And yeah, unfortunately for this treatment plant in Harnesporer it was too late, but maybe here in the shut-in wastewater treatment plant. We will have to see how the pilots are going here. So that is one part of the story I wanted to tell you. The other part is like my new hobby, anaerobic digestion. And in anaerobic digestion, I mainly focus on biogas production, but that could be as well volatile fatty acids. It just depends on where you stop the whole degradation process of your sludge. So anaerobic digestion of waste sludge. And to get, one of the aims in the Netherlands is to create less sludge, because the sludge needs to be incinerated, and incineration is pretty technology intensive and expensive, so we want to have as little sludge as possible. But actually in water treatment system, if you have the biological treatment, that is not really possible, because with the COD that you put in, you will create about 6 grams per liter of sludge per person today. Might be different if you have the saline water, like here in a bit more saline water as in Hong Kong, then you might be a bit lower than that. But this is for all the other biological systems that are on like low salinity, the sludge that you would produce. This sludge consists of living bacteria, but that's actually only a very small fraction, 10 to 15% of the organisms are in there. And then there's a whole lot of other junk in it. That can be higher organisms like worms and protozoa. It's partly carbohydrates and proteins that are in the extracellular polymeric structures. There's lipids. Of course, there's DNA, RNA also around from the organisms that died. There's a lot of humic matter in it. And there's these kind of stuff that come from the wastewater and is incorporated immediately in the flocks. So that is fibers, it can be cellulose, it can be plant materials, cell fractions, clay, precipitates, some sandy, silty fractions, heavy metal, hair. There's a lot of hair in sludge, which really frustrates my PhD students who have to work with real sludge because they have to get it out. It's a dirty job. But there is also organic micropollutants in it, so that is done very well. We want to take it out of the wastewater and it ends up in our sludge. And plastic. There's a lot of microplastics in sludge, from synthetic clothing, from scrubbing material, from tire runoff. In the Netherlands we don't have separate sewers, so the runoff from the streets it all ends up in the sludge. Well, to be able to get less sludge in the end and better dewaterable sludge, we could digest the sludge. So if we want to get the sludge production down, we should get the sludge conversion up, and luckily then also the biogas production per gram of sludge will increase, and that's what we like in this circular economy. If we get the conversion up, also we reduce the amount of our organics in the sludge, and then we will get our dewaterability up, which is then less volume of sludge in the end, which leads to lower process costs and also lower energy need for the final incineration. Well, actually this whole degradation of sludge starts with hydrolysis, and that's where my real research interest comes in. I want to understand how these big structures, these big molecules in sludge are hydrolyzed and cracked to be able for the next steps in digestion to be converted. So hydrolysis is actually breaking up large molecules with enzymes into smaller molecules that are easily further degraded into volatile fatty acids or biogas. And this can be enhanced by pretreatment of sludge, like the Combi system or other type of pretreatment, increased mixing in the digesters, and a different reactor design. So we're also studying plug flow digestion. So this is my current research focus, but I'm not going to speak about it now. So if you want to know more, then you can ask me later, because I would like to go back to this granular sludge. Oh, not back to there, back to the granular sludge. Because what we recently discovered is that the waste granular sludge in the Netherlands, so without pre-treatment, without pre-settling of the wastewater, so just with after sand and fat removal, we feed all the water to the Nereda systems in full scale. Then our waste sludge looks like this. So this is the small granules and the flocks that we want to kick out, but also a lot of fibers and other suspended material. And then the aerobic granular sludge from the reactor looks like this. So you have the big granules, and then you have this material that we actually don't like that much in between, and we remove that every cycle. And these are the washed granules. So this is actually what we call aerobic granular sludge. Keep this picture in mind, please. So what we did is, since I'm interested in digestion, we said, okay, let's digest these different fractions of sludge. That might be interesting, and actually it is. And we checked the composition of the different sludges, and we could see that the waste granular sludge, so the big granules, and waste activated sludge are pretty much the same in composition. Only the granules had a bit more lignin-like structures, but the rest of the composition was more or less similar. We did that with the Van Soest method for people who know it. And then we digested it, and then we were really surprised, because if we look then to the methane production throughout digestion, we can see that the waste aerobic granular sludge is actually pretty stable. So the granules don't produce so much methane anymore. And we think that is because these granules are for a long, long time in the reactor. So the inside of the granules, everything is eaten that can be eaten there. So it is that is stabilizing the granules as they're in the reactor. This is waste activated sludge, as we all know it. But then the the waste aerobic granular sludge, so the fluffy material that we wash out of the cycle every cycle, is actually pretty close to the primary sludge that we take out with primary selectors. So therefore, we can see that this highly nutritious material that we call primary sludge ends up in this flocculated fraction. And since it's washed out like every 2 cycles, it's not long enough in the Nereda reactor to be oxidized. So when we take it out, we can still digest it and come to a very high biogas production, which is very nice if you look into the resource recovery. Well, and then we have cellulose, which is our standard just to check if the experiment is really reliable. So our control. Well, thinking of that, we started to look a little bit more. Why are these granules so resilient to digestion? It was actually the next question. And then we looked to the granule size. And we could see that in the size distribution, this is activated sludge, and this is granular sludge. And the activated sludge is falling apart during digestion. That's something we know. So the particle size is decreasing. And then in granular sludge, we also saw it, but still, there was quite a large percentage of big granules that remain after 44 days of anaerobic digestion. So these granules are stronger. And we were thinking, what can that be? And we came to the conclusion that that is the structural EPS that is holding this granule together, that decreases slower in aerobic granular sludge than in waste activated sludge. And you could also see that from the proteins and the polysaccharide decrease during degradation. So then I come to some concluding remarks of all this research and experience. And the first one is that the aerobic granular sludge technology, this is what I said, it reduces energy, building material. And actually the effluent is even very good as to recycle and reuse in the end. You could use membranes for the last filtration step, and some tests also showed that these membranes, they hold very long. This primary sludge that you feed to a Nereda ends up in the flocculated fraction of the waste granular sludge, so not affecting the processes, but could lead to high biogas recovery. And this structural EPS is a biopolymer that's not easy to degrade, but it gives structure to the granules. And that can be recovered as a product before or after anaerobic digestion. And my research now continues on this structural EPS to see how we can crack it and make it biodegradable as well. Because at the places where you don't want to recover it, it would be nice to be able to make biogas or fatty acids out of it. But actually, my final concluding remark is that it doesn't really matter if a new technology is invented or research is done to solve a problem like footprint, that you want to reduce footprint, or that you want to increase biodegradation, or that you want to create struvite, because from all these new innovations, in the end, the circularity could benefit. So I would like to thank my team of PhD and postdoc researchers who actually make this all possible, because otherwise I could not have told you this new research. And I was requested by the embassy to show our online courses of the TU Delft, but they're actually pretty nice. And I'm working now on one on aerobic granular sludge as well, that will be released in January 2020, so I still have some work to do. And there is also an IWA World Conference that I'm organizing on anaerobic digestion, and that is from 23 to 27 of June in Delft in the Netherlands. So if you want to come to the spacious and green and quiet Delft, you're welcomely invited to register, of course. So thank you for the attention.